Okay. Welcome everybody to our CART Distinguished Lecture Series. I have the distinguished pleasure of welcoming and introducing Dr. Tim Roberts today, who is coming from CHOP in Philadelphia and the University of Pennsylvania, and will be presenting his work on stratification biomarkers for ASD, insights for multimodal imaging. We're really excited to have one of our first in-person and hybrid kind of virtual speakers for those of joining us in person and online. Just a few housekeeping notes to mention before we get started. This UCLA CART Distinguished Lecture Series is sponsored by our Tarjan Center, as well as the UCLA IDDRC and the UCLA Brain Research Institute. This is going to be in a webinar format, and so attendees may ask questions sort of throughout the lecture, and I would ask you to use the Q&A window. And we'll be recording this talk and posting it on the CART website shortly after. And so, as I mentioned, I have the distinguished pleasure of introducing Tim Roberts today, who holds the Oberkircher Family Chair in Pediatric Radiology and is the Vice Chair for Research in Radiology at Children's Hospital in Philadelphia, as well as Professor of Radiology and Linguistics at the University of Pennsylvania. Dr. Roberts received his bachelor's, master's, and PhD from the University of Cambridge in physics, and went on to do a postdoctoral fellowship in radiology in San Francisco before joining faculty at UCSF and then coming to CHOP, where he has really done some pioneering work using MEG, magnetic encephalography, to really characterize auditory signal processing in autism. And I've gotten to know Tim quite well over the last decade or so. He was actually on my thesis committee and an integral member of my PhD work, really trying to characterize auditory signal processing and translational biomarkers. And we wrote a couple papers and a book chapter together, and it's really fun to be back together. Tim's had an incredibly distinguished career. He's published over 300 scientific papers today, 30 book chapters, a large number of invited presentations and awards. I could go on. Really doing pioneering work in MEG in pediatric population, as well as MR imaging. And with that, we welcome Tim, and we're really excited to see your lecture today. Thanks for joining us.
Thank you very much, Mike. It's amazing that it was a decade or so ago, and you've gone on to bigger and better things, and I'm doing the same old stuff. I'd like to thank Dan for the invitation to come here, and I'd like to thank Monica for making it all work so smoothly. Thus far, it's not her fault if it stops working smoothly now, because everything's set up, it's all on me. So in the interest of time, I'm going to just fire away and get started. And the main idea that I want to do is to share some insights, I suppose, into the idea that we need stratification biomarkers for ASD. And I have some, disclosures that don't really interact, but I'm obligated to make them, including the intellectual property ones. So what I really want to talk about is this item at the top, defining biologically based subtypes based on multimodal MEG and MRI and MRS. But while I was thinking about stratification, I thought about some of the other things that we do in the lab and how they also actually play into or leverage the idea of stratification. So once we get through the main part of the talk, I'm going to spend a couple of minutes on each of these other topics, just to give a flavor of some of the other activities that we're doing, and also a more general idea of the need for stratification. So this isn't going to be news to anybody who's watching this, but ASD is heterogeneous. I think we all know that. And consequently, diagnostic biomarkers have struggled to be efficacious because they're trying to diagnose a heterogeneous phenotype. And I think something that's plagued the field whilst people have been having tremendously good ideas, is that most studies, certainly many of ours, offer group differences that look really promising, and you can certainly get your paper published in whatever measure it is that you're interested in. But then we always acknowledge the considerable overlap and recognize that this is probably not going to work at an individual level. So that's a bit disappointing. And one of the questions that we struggle with is, why do we continue to treat this heterogeneous spectrum disorder as a single entity when thinking about biomarkers? So the real goal, I think, at the moment is to identify some basis for stratification, to identify subpopulations which are within a group less heterogeneous. So how could you do that? Certainly you could look at the genetic etiology. The trouble is there are too many genes, there are too many subpopulations. And if you start including environmental interactions, perhaps it gets even worse. Granted, they may cluster, and I wouldn't say that we shouldn't pursue this. I think it's very worthwhile. But right now, it seems that there are too many subpopulations that way. Can we stratify based on clinical and behavioral phenotype? Absolutely. Can we manage based on that? Absolutely. But doesn't really offer very much mechanistic insight, what's going on in the biology of the brain. You can write papers about how conventional imaging like MRI can, usually with machine learning or something like that, can make some sort of diagnosis with some sort of sensitivity and specificity, but still doesn't offer very much in the way of insight. So what about taking multiple imaging approaches or a multimodal imaging approach? And I want to give one example of how we've had a go at this. There are lots and lots of other bases for stratification. I'm not saying this is the only one, please be clear about that, but this is an approach to studying the a variety of stratification. And it involves MEG, it involves MRI, it involves MRS. So I should probably explain what some of these things are. MEG is kind of the magnetic cousin of EEG. Basically, every electrical current in a neuron generates a magnetic field. The magnetic field comes out of the head. If you have an appropriate magnetic field detector, you can pick up that signal. You can actually, if you have enough of them, you can map the magnetic field. Once you know what the instantaneous magnetic field pattern is on the surface of the head, you can model it with some physics and identify the source of the magnetic field. Since you can do that in real time or on a millisecond by millisecond basis, you can effectively probe the activity of any region within the brain and ask essentially what's it doing in the way that you might do with electrocorticography or implanted electrodes. So we like to think of MEG as a 5-dimensional modality. It has a spatial component, where in the brain is stuff happening, a temporal component, when, and probably I'm going to spend most of my time on that today. And also we have a spectral dimension. We can essentially do a Fourier transform and ask the question, what type of activity is going on at any moment? So spatio-spectrotemporal insights. And here's a finding. So this is a child in the MEG helmet. And inside that helmet are 275 of these magnetic field detectors, whilst the child is experiencing a stimulus. In this particular case, it's an auditory presentation of a very simple tone. Most of the grad students, postdocs that come into my lab want to study really interesting sort of cocktail party things like metaphors, jokes, and at least sentences. And we haven't really moved far beyond beep, because it turns out there's a lot to understand about beep before we go on to the more subtle things. So you can see that the magnetic field pattern responds with these little bursts of activity, which we label things like M50, M for magnetic, 50 for 50 milliseconds. There's another one called M100 and M200. And what we have observed over the last decade or so is that these components, the M50 and the M100 in particular, tend to be delayed in ASD. They tend to be delayed by 5 to 10 milliseconds. And it tends to be very variable. But nonetheless, there are robust enough group effects that we think something is going on in the timing of the evoked response. And if you think about that, that means that the time it takes to process the sensory environment is delayed. That's just the first thing that the brain has to do. So if every other station of the brain is similarly delayed, you can see how that could cascade into a terribly challenging processing of life, of the experience. So we're going to try and get a handle on this M50 delay. To note, we've been studying this for a while in different labs and with different hardware, and looking both at the M50 and the M100 component. And I'm going to switch between those throughout this talk, primarily because the M50 works better in young children, the M100 works better in older children. Some other labs have been doing this as well in New Mexico and San Francisco and Vanderbilt. And one of the things that was observed is that maybe the latency of the M100 has at least some sort of severity correlate. So if we take the SRS as a measure of autism symptom severity, and of course there are other measures, we can see that the latency is longest, is quote unquote worst in the cases with the higher SRS score. So that's indicative of difficulties processing the sensory environment, or delays processing the sensory environment. But you'll also notice there's an awful lot of scatter there, which is typical of many of the findings, which is that you get a trend, you get an effect, but you wonder if that could actually be meaningful on an individual basis. And in any case, although clinical correlation like this is nice, I don't think it justifies the term biomarker. There's a bio in biomarker, and at this point, we don't have any biological insight into it. So one thought that had occurred to us is the delay, this M50 delay or M100 delay, simply due to poor conduction velocity along the auditory white matter pathway? So can we image conduction velocity along the auditory white matter pathway? Well, we can sort of image the white matter of the auditory radiations using this technique called DTI. And the arrows point to the whiskers of the auditory radiations. You can, in fact, go all the way from the brainstem to the thalamus to the cortex, but that last whiskering range is what we focus on, and it allows us to use this technique called DTI, which is an MRI technique pioneered many years ago, and then has been interpreted as imaging the fibrousness of white matter. And at some level, the white matter integrity relates a little bit to the axon diameter and to myelination and things that would generally physiologically speed up conduction velocity. 30 years ago, I made these images of some celery to show that you can also image fibrousness of celery. I haven't really been able to use those enough in my life, so there they are for your amusement. The white dots there are the bits that get stuck in your teeth. So when we looked at the white matter of the auditory pathway and the quality of the white matter indexed by a rather simple parameter then called the fractional anisotropy, we noticed that that matures with age, that the M100 latency shortens with age, and that was pairwise truish, which allowed us to come up with a biophysical hypothesis that maybe the diffusion numbers that we can generate relate to the maturing axons of the white matter, and that maturation leads to an increased conduction velocity, and increased conduction velocity leads to a shorter latency. And we got close to thinking that was a real biophysical hypothesis. Then we tried it in autism, and we noticed that while the white matter was atypical as a group in autism and matured atypically, there was crazy scatter and variability. And in fact, this pairwise association between diffusion properties and evoked response latencies didn't really hold, leading us to conclude that, it's a pretty strong conclusion, factors other than white matter conduction velocity impact the maturation of the auditory evoked response. So like there's something else we hadn't thought of. So what else could there be? Well, the other thing that you have between the ear and the auditory cortex are synapses. So maybe we should look at neurotransmitters. This is, taking us a little bit more towards biology as well. And we use a version of magnetic resonance spectroscopy. Now, if you look at the top right, there is a spectrum, a typical MR spectrum, and it has those three characteristic lines in them. And this is what you'll see on a clinical scanner. One of them is for N-acetyl-aspartate that has something to do with neurons, but we don't know what. One of them is creatine, which has nothing to do with much, and one of them to do with choline. And if it's low like that, that's good. You don't have a brain tumor. And so that's, I wouldn't say it's boring, but it's not incredibly rich in information. But it turns out there's a cleverer thing you can do with spectroscopy, and I certainly don't have time to go into the details, where you can effectively do two slightly different spectra, subtract them, and take advantage of something called J coupling, and basically make all of the boring peaks go away, and all you're left with is GABA and glutamate. And so that's what we do. The devil is in the details. There's all sorts of little tricks that you have to do: macromolecular suppression, correct for field drift, exclude transients that are associated with motion, lipids, get the subtraction right, etc., etc. But when you do this, you have a modality that at least gives you access to the inhibitory neurotransmitter GABA in a somewhat quantitative way. And that's relevant because now 20 years ago, John Rubinstein and Mike Mesnick at UCSF had or proposed this excitation inhibition model of autism that would predict depleted GABA in the brains of children with autism. And they pretty much got this idea from the co-occurring epilepsy that was known to be common in ASD, and the glutamate to GABA imbalance in epilepsy. And then we had the idea that, well, if you've got atypical inhibitory interneurons impinging on pyramidal cells, if something is broken either with the neurons or with the GABA, then it might upset the local circuit activity that we could pick up with electrophysiology. Thanks to Bill Gates, not the Microsoft one, my Bill Gates, for this cartoon. And Bill did some work a while ago showing that you could measure GABA like this in typically developing individuals and kids with autism. And indeed, you could see depleted GABA, particularly in the temporal lobe, auditory cortex, a little bit in the frontal lobe and motor cortex, and not so much in the visual lobe. And then one of our grad students, postdocs, Russ Port, was able to show that what happens is that this GABA during development leads to an increase in gamma oscillations, which are a hallmark of intact local neural circuitry. So that's the inhibitory interneurons impinging on the pyramidal cells, forming a nice circuit, making a nice oscillation in the gamma or 40 hertz range. And when you do that, as during development, if there's sufficient GABA present, then this gamma oscillation goes up as the brain develops. And by adulthood, you have fairly strong gamma oscillations. What we found in a cohort of ASD was that there was no such clear relationship between GABA and gamma. And consequently, in adults, gamma findings tend to be reduced. That allowed us to sort of revisit this EI hypothesis through through our sort of macroscopic and if you like, far field approaches. The problem is that the EI model is often correctly considered simplistic, or sometimes even just wrong. If it's derived from the observation of co-occurring epilepsy, why don't all individuals with autism have seizures? Arguments like that. And so, of course, this is another argument for stratification. And presently, our prevailing thought is that it might well be a useful model for some individuals with autism. And moreover, that might provide a rational basis for defining a subpopulation who indeed seem to have this particular imbalance in biology. The GABA levels may matter in some individuals, and that has all sorts of implications for treatment. So the math folks in the lab said, well, why don't you come up with, if you want to try and account for that M50 latency, why don't you use a linear model? Latency is something to do with diffusion, something to do with GABA, something to do with age, maybe some other stuff, some errors, cross terms. Sounds complicated to me, but sorry, it's not even as simple as that. So what we actually did, and I'd like to thank Jeff Berman and Luke Bloy for helping me a lot with this, is we found that in typically developing children, the M50 latency is very well predicted by the thalamocortical white matter diffusion properties and age. 52% of the variance accounted for, that's really quite a lot for a simple model in biology. However, when we applied this model to children with autism, a population of 100 plus children with autism, many of those individuals fell somewhere in that typically developing range, suggesting they may have immature white matter, but they had appropriately immature evoked response latencies, so they were still conforming to the pattern and were perhaps just delayed. But a population of about 15 to 20% were clear outliers to the model. They lay outside the normal, the typically developing confidence interval, and seem to have unpredictably long M50 latencies. So what about this subpopulation we've defined, the ASD with unpredictably long M50 latencies? That sounds interesting. Was there any age or sex dependence? No. Were there any clinical sequelae? No. Didn't seem to differ on ADOS, didn't seem to differ on IQ, didn't seem to differ on SRS, didn't differ on anything we could measure. So you could ask, is there any relevance at all to defining such a subpopulation that doesn't have a clinical correlate? And the answer is yes. In fact, maybe that's even the point, because if you define based on clinical groups, then we found a nice correlate, but we haven't done anything additive. But what we did find is that these individuals who were outliers and had unpredictably long M50 latencies had atypically low levels of inhibitory neurotransmitter GABA. So the main significant finding was that GABA became rate limiting for the M50 latency in this subpopulation of outliers alone. It didn't matter in the whole, in the general model. Once you have enough GABA, having a little bit more doesn't do anything for you. But in this subpopulation, who had very low GABA levels, that GABA influenced their M50 latency. Wow. So we have a subpopulation with a biological difference that you wouldn't have been able to observe clinically. There were no clinical sequelae, and yet two individuals who have very similar clinical performance, one has low GABA and one doesn't. Why would that matter? So in parallel with this, we conducted a little bit of a, not a clinical trial, something like a clinical trial experiment, where we acutely administered a GABA-B agonist, arbaclofen, to 25 or so adolescents with autism. And we did MEG before and after, before and 1 hour after a dose of placebo or low dose or high dose. And we asked the question, does anything we can measure in MEG show any response to the drug that isn't random? So I'm not going to show all the results, it's in the paper, but you can look at the placebo, for example, and you see that there are random changes in the M50 latency, but none of them are statistically significant, defined for us as a z-score of more than 2.57, so the 99th percentile, so a 1% chance of it happening by random. Placebo didn't do anything. Most of the measures that we tried didn't show any particular pattern, except the M50 latency at one particular dose. And you see at the top here, about 7 or 8 areas of marked as blue, suggesting that they exceeded the threshold of 2.57 Z's. So you would expect maybe 1 in 100 by random chance, and we're getting 7 in 25, suggesting that about 30% of the population had an acute response to this drug. So that tells us two things. It tells us that the M50 can be responsive, but it's only responsive in some people. And if I was running a clinical trial, those are the people that I would want in this trial, the ones that at least show evidence for biological activity. I don't know if that ultimately is going to lead to clinical outcome improvement, but at least it shows that something's happening biologically. It's also, by the way, a neat way of doing dose finding, because it turned out to be the lower dose that was more efficacious than the higher dose, funnily. So I couldn't come to UCLA without giving a nod to Mikegandal, who so kindly introduced me. And I just wanted to point out that he made the astute observation a decade ago that these things that we measure in children are rather absolute and quantitative and objective. And could you apply that to rodent models of autism? And in this case, he was working with Steve Siegel and looking at the valproate model, and showed that very analogous findings were observed in terms of the latency of electrophysiological auditory responses, showing something like a 10% delay, and also that the gamma oscillations that we believe are associated with depleted GABA paralleled again between the human and the mouse. And I thought this was just sensational work. So I want to bring that to everyone's attention and thank Mike again for allowing us to work with him on that. Okay, so in a way, that's what we've got on stratification. We've got the idea that that most things, most measures, most metrics don't work for all children with autism. And again, that really shouldn't be much of a surprise. But what we've been able to do, and it wasn't that easy in this example, we've been able to define a subpopulation and then identify that the subpopulation that we defined as having atypically, unexpectedly long evoked response latencies beyond what you could predict from white matter, well, they were characterized by having atypically low levels of the neurotransmitter GABA. And for me, this is beginning to allow me to say that the M50 latency or the M100 latency is a biological marker, a biomarker, because we have these sort of biological evidences. It's not going to be a diagnostic marker because it doesn't work in everybody, but maybe it's going to be a stratification marker because maybe in those individuals for whom this is an identifying marker, there might be a treatment that, like arbaclofen, that is tailored to that particular biology. So you can take a breath there. And now I'm going to talk about 4 vignettes, probably 5 minutes each, talk about some of the other things that we're doing in the lab that I think are very important to a comprehensive study of biomarkers in ASD. And the first one is to recognize that the autism spectrum is a very wide spectrum. And especially when we're thinking about language, we have a significant proportion of individuals with autism, children with autism, who are minimally verbal or essentially nonverbal. And most of the time, these individuals are not included in research studies. And there's and the reason for that is because it's quote unquote too difficult. And that really shouldn't be a barrier for any of us conducting research. And the reasons for doing it are so important. The first one is scientific. How do we know that any of the things we observe will generalize to the whole population if we don't study the whole population? So I think, you know, that's good science about generalizability. But I also think there's a societal role here. You know, if we don't include constituents from the full spectrum, we're not giving them a chance to contribute, you know, what they can to scientific endeavor. And for many of the families that we've spoken to, they desperately want to be allowed to participate in this process and to contribute to science. So I think studying minimally verbal, nonverbal, and generally severely impaired individuals is very, very important. That said, it's difficult. So very fortunately, one of the neuropsychologists in our team, a clinical neuropsychologist, Emily Kushner, came up with this protocol for enabling MEG scanning of children with limited abilities, she called it. And it's a mixed idea. There's some technical components and there's some behavioral preparation. There's a lot of ABA training that goes into this and an adaptive behavior of the scanning support team. But there's also the idea, very much so, that the parents are involved from weeks before. We do a lot of preparation and desensitization. Nothing groundbreaking, but when you do it all right, you find out what are the reinforcers and you color code the various rooms, and if a particular individual has a particular reinforcing preference, then we can certainly use that and try and encourage behavior. A lot of visual supports, very much individually tailored experience. Everybody's visit to the MEG lab is a little bit different and it's special for them. Then when they get there, there's a whole bunch of technical things that we put together, like short paradigms, passive paradigms, motion compensation, obviate the MRI requirement, and just use mathematical modeling of that. And all of this was not something we came up with on our own, but we literally had focus groups and consulted with various stakeholders and did some qualitative research to find out what would maximize the experience. So between Emily and Mina Kim, who's a BCBA in the group and is now actually on the neuropsychology faculty too, we put it in practice. And indeed, we were able to record M50 evoked responses in minimally verbal children with ASD with remarkable success rate, I think 75% or something like that. And as a measurement scientist, one of the things we noticed was that there's a reward for doing this, which is that the effect sizes get bigger. So indeed, the delays that we see in minimally verbal children with autism are in fact greater than those that we see in verbal children with autism. So although it's a tremendous amount of work and endeavor, MEGPlan enabled us to be able to do really broad spectrum science, and we're very, very excited about that. Just as an example, we haven't talked about the mismatch field at all, but that was another measurement that is commonly done in electrophysiology, where we look at change detection response. That is, if I'm saying and then I say ooh, there's a characteristic brain response that says, hey, that's a little different. And that's important because it corresponds to the changing syllable in a word. And if that response is delayed, you can imagine a sort of, again, a catastrophic cascading. And we found that in the minimally verbal children with autism, that response was delayed even further than the verbal children with autism, in whom there was already a delay associated with their degree of language impairment. So really, we got the strong idea that the mismatch field relates to language. There was also some hemispheric asymmetries that we can talk about. Another measure that we've looked at that's getting a little bit closer to something that's a little bit more interesting than R's and U's. We did an experiment called Lexical Axis involving words and plausible non-words. So the canonical example of that is block, that's a word. Blick is not a word. It has many of the structural elements of block, but it's blick. And if you present these words, you can record the evoked response. But now we're not interested in just the timing of the evoked response. We're looking at that information in the spectrum of the evoked response. So it's a little hard to interpret these, but the blue blob is basically a period of oscillatory activity in the range 5 to 20 hertz between 200 milliseconds and 1 second post-stimulus. That's a lot, but that's a very specific measure, and we can extract that measure using MEG because it has temporal resolution and spectral resolution. And if we integrate that blue blob, it turns out that the blueness of it indicates the degree of language ability. So if you have very typical language ability, you will have a strong blue blob, and as your language is impaired, the blue blob decreases. So Luke Bloy published this as a robust estimator of language, acknowledging again that there's a lot of scatter there. So there's interesting group level effects or interesting trends, but I'm not sure how useful this is on an individual basis. And this perhaps needs further stratification along the lines of what we were talking about before. We then extended this to children who were minimally verbal and with severe cognitive impairment. And of course, to understand that a bit more, we recruited a group of children who did not have an ASD diagnosis, but were similarly cognitively impaired. So they had also very low IQs. And we're able to find that we could still just about see the blue blob, but of course it was diminished because the language ability was lower in these individuals. But even at the minimally verbal, non-verbal end of the spectrum, we saw that the blue blob integral did show a trend where the more language ability that was present, we would see a slightly increased blue blob, suggesting that this is truly a marker of language ability that extends all the way down to non-verbal. And that if we could use that as an index of, we might be able to use it as an index to study the restoration or recovery of language ability, whereby any efficacious treatment would be expected to increase the intensity of the blue blob. So that's what we put together here, then basically a battery of 3 experiments, the M50, which we saw similar delays in the non-ASD intellectual and developmental disabilities cohort. So we feel that this is probably, the M50 latency is probably, at least in this population, dominated by general cognitive ability. It's a better predictor of general cognitive ability. The MMF certainly had something to do with general cognitive ability, but also was strongly influenced by language. And then this ERD, the blue blob, really differed between the minimally verbal children with ASD and the IQ matched cognitively impaired controls, suggesting it's really their language differences that were dominating the ERD, because whilst they were matched on IQ, they were not matched on language ability. Taken together, those three form our 15-minute electrophysiological probe battery. Just want to mention that we have looked in a sort of genetics first way at a couple of genes associated with autism. One's a copy number variation, 16p11.2. We have the cohort of boys with 47XYY, so a whole extra Y chromosome, and other things like NF1 that I don't have time to talk about. And I just wanted to make the point that this M50, or in this case, M100 delay that is implicated in ASD and is given to us by ASD actually shows up as a correlate of genetics and gene dosage, in the sense that children with 16p11.2 deletion showed delayed M100 latencies, whether or not they had an ASD diagnosis. And when you had extra genetic material in the duplications, we actually saw no delay or possibly even a facilitation in the latency, suggesting that the relationship between genes and physiology is tight. Gene dosage matters in predicting the electrophysiological response. Similarly, just an example to show that boys with 47XYY who have a high rate of ASD diagnosis also showed delayed electrophysiological responses associated with perhaps the gene dosage of genes on the Y chromosome. And at this point, our follow-up work is investigating neuroligin 4Y as a possible gene expression correlate of the magnitude of the delay. So genetics and electrophysiology coming together in both of those studies. And as I used to say on Monty Python, and now for something completely different. And I want to acknowledge that the next few slides have hopefully some commercial implication. Given all of the science that's gone on in the first part of this talk and the recognition that we really don't know everything, we don't even necessarily know very much, is there anything we can do right now? And so for me, this is a complementary approach, doesn't replace what we're doing in terms of mechanism and mapping genes onto brain and ultimately brain onto behavior. But how can we help? How can we actually help children with language difficulties right now? And I would remind you that I made a disclosure about a patent that my colleague in linguistics and myself own. And this is for something called a language aid. And this is something is in the infant stages of commercialization at the moment, but is basically in the form factor of earbuds, and it addresses language difficulties like this. And again, the reason that it's okay to talk about this is because there's clearly a need for stratification of the ASD population. I'm not saying for a moment that this works for everybody, but one thought is that speech processing in at least this subpopulation of ASD can be complicated by hyperattention to acoustic details. That is, not tolerating tiny acoustic differences in the speech that are irrelevant for the meaning and are typically glossed over to form a unitary object perception, but are not and are retained in this subpopulation of children with ASD as separate, unique tokens. And to try and represent that, I've taken the word cat and imagine 100 utterances of cat. They could differ in color, in font, in size, in case. And when the typically developing individual experiences all of those flavored or colored or fonted cats, a single representation of a four-legged furry feline exists. And that's a way that we abstract meaning from speech words, tolerating subtle differences and saying that's not relevant for the meaning. We're talking about a cat, not a pink lowercase times cat versus a different, no, we're talking about a cat. And that helps for communication. It speeds up the communication of ideas. It saves a lot of memory in the lexicon. So the thought here is that digital signal processing can potentially remediate this. So language aid is really like a hearing aid for language that will help eliminate those extraneous distracting details that are difficult for some children with autism to ignore. Now, the actual algorithm may need personalization. It may not be the same approach for everybody. It's not going to be simply, well, you could just filter this. So what is it that we need to do? Well, we think that that blue blob language index may help tune the algorithm and thus personalize this intervention. Very much akin to when you go to the optometrist and they pop in one lens after another, and eventually you empirically determine what your appropriate prescription should be. We might do the same with one filter versus another filter versus adding some noise until we maximize the blue blob. Then tweak the digital signal processing in the earbuds, and then give this personalized modification to the auditory stream to the individuals in the hope that that enables improved language acquisition, improved communication in life. Again, this is the example of that. The complex things which are speech lead to very different brain responses. And if you don't do any processing, you have all of these multiple representations. But if we can do a sufficient processing, we may be able to restore our language index, and perhaps that corresponds to distilling all of those multiple flavors of cat into a single, albeit decolored, grayscale cat. So the main idea is that we think that the acoustic manipulation, when tailored appropriately, can restore the brain response. And we believe that that would be useful for subsequent clinical improvement. Just like we believe that the test dose of arbaclofen tells you that you have a biological response, that you would then need repeated doses to have clinical benefit. This is in the same way saying we can do something that is modifying the brain and making the brain more receptive to these language stimuli, and hopefully over the course of weeks and months, that would improve clinical language performance. So that's just an idea. It's very different. One more thing, you can do this in the sense of a priming, and we've demonstrated in a handful of individuals that you can show that speaker voice differences are not tolerated in some individuals with ASD, but by a simple filtering in this case, to take away pitch effects, so that the male versus female differences in speech, we can recover the blue blob in this experiment. So we think there is science to support this. Of course, more work is needed. And the last thing I want to talk about in the last couple of minutes is back to the idea that, well, even though we need stratification biomarkers, what about infant predictors of ASD and indeed intellectual disabilities? Can we go for early diagnosis? And we're recognizing there are many cohort studies looking at high-risk individuals with autism, but I've got a point here that I think speaks a lot to the stratification and a lot to the scatter that we've seen across many of these plots so far. But in order to do infant studies with MEG, you need an infant MEG scanner, because the physics of MEG says that the sensitivity of the coils depends on the distance from the brain to the detector, and infant heads are smaller. And if you put them in an adult or large child scanner, you have diminished sensitivity because of all of the wasted space that the magnetic fields are decaying in. So we thought, let's go and get ourselves an infant MEG scanner, except they didn't exist. So we had to design and build one. And this was a few years ago that we were able to build this guy called Artemis. And here's some data from that, from Heather Green, who's a postdoc in the group, and Chris Edgar, who's one of my faculty colleagues. And the thing to notice here, there are multiple little points. The blue dots are 70 or so low-risk infants with autism, infants with low risk of autism, so essentially typically developing infants. And you notice that across the first 2 years of life, the M50 latency does in fact decrease, as theory would have it, but with tremendous, tremendous scatter. And that's because babies develop at individualized different rates. So if you take individuals with high risk for autism, that's the red dots, mostly they are falling within the scatter of the blue dots. And you would be tempted to say, well, it doesn't work. There's too much scatter for an individual red dot to be diagnostic. And that's probably true. But if we take an example of looking at the longitudinal developmental trajectory in green of a handful of typically developing children, what you notice is that whilst the snapshot values are scattered all over the place, there is a strong similarity in the slope of these lines. The developmental trajectory looks very similar, and most importantly, shortening over time in the green lines. That is to say that a lot of the inter-individual differences can essentially be taken care of by asking not the question about the latency, but the slope of the latencies. How many milliseconds per year are you maturing? And that's really a general approach that I think points to the need for longitudinal studies to really compensate for inter-individual biological differences, using your baseline measure as its own control, if you like, statistically by treating subject as a random effect. But here's the interesting thing. Two of these red dots are from the same individual scanned at intervals. And what you notice about those left hemisphere and right hemisphere lines, but particularly look at the right hemisphere, is that the developmental trajectory of the M50 latency in this individual from 12 to 24 months doesn't decrease like the green lines. It's flat. In fact, it might slightly increase. And so whilst you never would have identified the red dot as being atypical of the blue dot distribution, you would readily and easily say, well, that slope looks different. That's not maturing in a typical way. And whilst this child doesn't have an autism diagnosis yet, developed milestones indicate that this child is definitely performing at the very lowest end of the range. So we think here that longitudinal designs will be critical to help account for the variance that otherwise sort of smothers any instantaneous finding. And so does that mean we might have to do 2 scans in order to make a diagnosis? Yes, we might. The last thing I want to say is we're now taking this approach not for the purposes of ASD diagnosis, but rather to look at the prognostic outcome in terms of what's the flavor of the deficit. Is it going to be a general cognitive, going to be more of a language deficit, something in a specific domain? And we're looking at children with genetic risk of an intellectual developmental disability. This is our IDDRC project. We're going to take infants at 6 months with some risk of poor outcome, follow them until they're 36 months. At the same time, we're going to take individuals at 36 months or 3 years and follow them till they're 6, when a more robust estimation of their clinical outcome can be made. So it works like this. We might have a genetic risk, whether it's a single gene like Fragile X or a CNV like 22q or any other known genetic risk. Also, other risks that are of not yet known genetic etiology, but have been determined to be sources of risk, either by backward looking through the electronic medical records or through pediatrician observation. We're going to apply our battery, remember the M50, the MMF, and the word-related blue blob, ERD, which are differentially sensitive to cognitive and language abilities. We're going to scan with our MEG scanner, Artemis, and model that battery of results into a prediction of outcome, which may be in form of diagnosis, but is more generally going to be in the form of a profile, a severity profile of whether these individuals are more likely to have a general cognitive versus a language-specific deficit. It's not the only form of stratification you can do, but again, this is a sort of a thematic example for the day. So I hope I've introduced some of the things we can do, some of the difficulties when you really get into the weeds with this, but that there's a need for biologically based stratification that might actually speed up pharmaceutical interventions. but we also have to recognize that there's a lot to be learned from genetics, genetic risk factors. There's a critical importance to sample the whole spectrum and include individuals who are severely impaired and for whom some of the subtyping might be most beneficial. I think there's a role for infant predictors, again, using the same battery. Let's not try and predict a clinical diagnosis so much as predict real functional outcome measures that will matter on a daily basis. And then while we're doing all of this basic science, let's work on intervening to do something in the here and now. For example, the language aid, again, guided by the brain responses and the learning from these other techniques. So, of course, there's a ton of people that have contributed to this work over the years, including Mikegandal. So I'm deeply grateful to all of these individuals, my colleagues in the sort of physical sciences, psychology, psychological sciences, radiological sciences, and a ton of coordinators and graduate students and postdocs who have really made this endeavor work. And to the funding agencies that have supported it, thank you very much. And to you for your attention. Thank you very much.